Today we'll go over the specific bacteria that are on the microbes and agents list. So before we talk about all these bacteria that cause diseases, it's good to know that they're what are called opportunistic pathogens, and this just means that these microbes essentially they can be present within our body to some degree even when we're not sick, but it's just when, let's say, our immune system gets thrown off or our microbiome gets messed up, that's when these bacteria can start to kind of flare up and mess with our bodies and cause disease. So just a good thing to note that they're not always going to cause disease. It's only when our body's kind of off kilter, so to say. So first, E. coli, of course, you've heard of this one. So it's gram negative. It's a bacillus. If you don't remember what those are, I'm hoping that you go review those terms. And they're well known for being enteric bacteria, or a bacteria that are in our digestive system. They're also the most common model organism in the lab. This is just a concept called reductionism, where essentially we can use E. coli, which are much simpler than humans, but we can still use E. coli as a model because even across life, right, there will be mechanisms and traits that are very similar all across life, even though E. coli are like much simpler than humans. So we can use them to test stuff that we might not be able to test on like actual humans, right? And again, E. coli, like I said, about the opportunistic thing, right? They're present in very high numbers in our body normally, but there are some specific strains that can cause disease whenever our immune system is off or other factors. And strains, right? This word strain you've probably heard about. It's essentially like it's like the way that all dogs, domesticated dogs, are technically the same species, but right, they can look very different, act very different, have very different traits. So that's the same thing with E. coli, same species but very different traits between different members of the species. So, for example, we give this classification of O colon H. So essentially, what that means is, right, the gram-negative bacteria, they have that O antigen, right, on the lipopolysaccharide in the outer membrane. This sugar thing that sticks out of the outside of the cell. So depending on what the composition of that O antigen is, and what the composition of what we call the H antigen, which is on the flagella. We just give a classification for different strains. So, for example, you might have seen in the news O157, H7. This is a very famous E. coli strain that caused several outbreaks in the early 2000s and early 2010s. So, so this is just a naming convention that you might see. So it's good to be familiar with that. So since there's a lot of different strains of E. coli that can cause disease in different ways, we'll kind of do a brief overview of the different ways that E. coli are implicated in human disease. So some strains, they can use, if you remember Fimbriae, right? Fimbriae, they're kind of like pili. There's those like extensions that kind of stick out of the cell, but they're pretty short, and there's kind of a lot of them located all around the cell usually. So some strains use those to cling to the cells in our intestines, and what we call the epithelium or epithelial cells. These are just the cells that are on the very outside layer of our intestines, and they can also secrete or kind of release what are called enterotoxins. These are just compounds that are very toxic specifically to the digestive tract. And then some other strains, right? They can use they use what's called an intimin protein, and this just is like a very sticky protein that again helps them stick to the inside of our intestines. And they also secrete enterotoxins, and one that you might see is called a Shiga toxin. Not really important to know, just in case you see that name somewhere, just to be familiar with it. So all these strains, right, E. coli, right, they cause diarrhea, and which will also lead to fever, dehydration, vomiting, just basically intestinal problems. So there's no specific treatment since there's so many different strains, but usually just rest and home recovery is sufficient to treat this disease. Right, next let's move to Staphylococcus aureus, or S. aureus, right? So this one is gram-positive. It's a coccus, as the name suggests, right? The staphylo prefix means that These cells are usually located in kind of clusters or sheets, as you can see on the right side. And they're known for causing, of course, staph infections. And staph infection is kind of a general term for infections that happen by this bacterium in different a bunch of different parts of the body. Also notable that Staph aureus is very resistant to antibiotics, and it evolves quickly, and we'll talk about that in a second. So after you get Staph aureus, right, you can get it, from contact with someone who has it. You might just get it from your environment, right? And there's Staph aureus all over your skin right now as well. So if you get like a cut, then some of that Staph aureus might kind of sneak into that cut and start infecting it. So a lot of different ways, but essentially after infection, S. aureus, it has many different toxins that it can kind of release from the cells, and these in various ways help it evade the immune system that's coming to attack it. So some cause the immune cells to actually lyse or die off, while others inhibit phagocytosis. If you remember, phagocytosis is when, for example, an immune cell will come and kind of swallow up a bacterium and then start destroying it by kind of shooting a bunch of enzymes at it that kind of break it down. So it can inhibit that process, and some other of the toxins can allow it to kind of penetrate into host cells and insert themselves inside. So again, the point is, S. aureus is very crafty. And like we said, it evolves very quickly, and it has already become pretty much overall resistant to penicillin and methicillin, which are two common broad spectrum antibiotics. Again, those are part of the beta-lactam class, right, or the cephalosporin class, and we talked about that in my antibiotic lecture, so make sure to review that if you don't remember. So essentially, so you can see this chart on the right side, right? As Staph aureus developed resistance to penicillin first in the 1940s, and it did that by acquiring this gene called BLAZ or BLAZ. So this gene, right, it codes for an enzyme that's called a beta lactamase, right? Remember, penicillin is a beta lactam antibiotic, so a beta lactamase kind of attacks penicillin and breaks it down. So these bacteria with this gene, they can basically break down all the penicillin that's getting shot towards them, so they kind of, they can survive because of that. And then you can see later on, right, methicillin, the second antibiotic, right, similar to penicillin but slightly different mechanism. And also, it's notable it's good to know that a lot of the mechanisms are super well known. So you might have two drugs that theoretically should have very similar kind of functions in targeting a bacterium, but in reality they might act very differently, and they might differ significantly in their effectiveness. So just something to note, just because how complex the biology is, and are the what we call pharmacokinetics, right? There's a whole field that's dealing with that. So just to know. But yeah, so methicillin resistant Staph aureus, or MRSA, you might have heard of this in the news. It uses what we call MEK-A, which is a gene. Essentially, it codes for an alternate form of a penicillin binding protein. If you remember, PBP, penicillin binding protein, these are the guys that go and kind of keep building up the cell wall and kind of maintain it. So, right, those are supposed to be the targets of antibiotics like methicillin. But now, since this MEK-A gene is making a different PBP, now methicillin can't recognize and bind to that PBP to inhibit the cell wall synthesis. So essentially, it can keep making its cell wall and function as normal. So yeah. And this vancomycin, this also attacks the cell wall of the bacteria. This was thought to be kind of a last line of defense, but even resistant to it has risen in the past few decades. As you can see here, we have Visa and then Versa. Visa is vancomycin intermediate, so it kind of halfway resistant, and then vancomycin resistant is Versa. Yeah, so now let's look at the details of a staph infection. Right, it's a broad term, as we said, for any infection by Staph aureus anywhere in the body. The severity can range from just slight skin irritation to even significant heart damage that can be life-threatening, such as what we call endocarditis, where the bacteria infect the inner lining of the heart and start messing with that. So at the skin level, you can see these pictures here, right? Staph infections, they can result in just simple, irritated rashes, blisters with like pus in them. And then more severe infections, we call bacteremia, where the Staph aureus, it spreads through your bloodstream and gets to different places throughout your body. That includes the heart damage that we talked about here, and it can also go into the bones, muscles, spinal cord, etc. So treatments, since Staph aureus is so resistant and so fast evolving, we need to use specific antibiotics for these. So tetracyclines are one class of antibiotics that seem to work against most strains. And then daptomycin. Oh, and if you remember, tetracyclines, they bind to the 30S ribosomal subunit, so they inhibit protein synthesis in bacteria. And daptomycin, this is one that we haven't talked about. It's a lipopeptide, so it's a protein that has a lipid component that kind of mimics the lipids in the membrane. So what it does, it kind of inserts itself into the bacterial membranes. As you can see, there's a red thing here. Then it kind of aggregates, makes these big clusters that break up the membrane, and that kind of that can kill the bacterial cells. And since we talked about all this resistance spreading to antibiotics, right? The reason or the way that these resistance happen is first through mutations, but then of course once you have like a plasmid that has, for example, the BLAZ gene, right? Then you have horizontal gene transfer that can occur either through conjugation or transduction that we talked about previously. So just a good review there. Now let's move on to rickettsia, I don't really know how to say it, but yeah. So this guy, it's an obligate intracellular, which means that these bacteria can only survive within host cells. They can't survive just like on the ground or in the environment by themselves. They're also gram-negative, and they're something we kind of didn't discuss, but a coccobacillus. So this is just something that's intermediate. It's almost spherical, but it's also not completely ovoid in shape. So it's kind of like an intermediate between circular and oval. These bacteria, they reside in ticks normally, and they can be transmitted to us through tick bites into our bloodstream. So essentially, let's say an infected tick will bite a human. The bacterium kind of spreads, and then it likes to invade the vascular endothelial cells. These are just cells that surround our blood vessels. So you can see that here you have this purple blood vessel, and then you have these red stained bacteria kind of residing within that tissue. So this infection, of course, and you'll see this is a common theme. Anytime you get an infection, the immune system kind of goes crazy, and it's as the immune system's trying to attack the bacteria, they produce what are called inflammatory cytokines, which are just molecules that essentially cause inflammation in the surrounding tissue. And so essentially infection will always lead to inflammation. And since this tissue, right, the tissue that is infected is surrounding the blood vessels also can become more permeable, since there's a lot of inflammation around here, which of course you don't want your blood to start leaking out into your other tissues, so that's not good. So the notable thing here is that this bacterium has a unique mechanism that it can use to move from cell to cell within your body, and this is called actin-based motility. So what it can do is it essentially hijacks the cells, like our cells, and what it does is it'll start using our actin protein, right? Actin is normally used in the cytoskeleton in our bodies, so within our cells, it kind of makes this framework, kind of skeleton thing that kind of supports the structure of our cell and maintains the rigidity. But it can kind of take some of that actin protein and start building a filament, multiple filaments at one of its ends, like this. As you can see, this actin has been stained red and the bacterium has been stained green here. So it assembles what are called nicknamed comet tails. And so once it has these long actin tails that it derived from our cells, it can then use those tails to propel like a comet. They can propel themselves forward, and they can kind of shoot themselves into nearby cells. So this is kind of a crazy adaptation that they've evolved, and this is actually exhibited in several other species of obligate intracellular parasitic bacteria, such as this one here. Yeah. So, and then of course, right, once you're infected, and then another tick that's not infected can come bite can come bite you. Then it'll pick up your blood, and it'll probably pick up some of the bacteria as well. Then it can go off, and then it can go bite someone else, and then you can see how the cycle will continue to progress that way. And yeah, ticks. Another notable thing is that ticks, female ticks that carry this bacterium are rickettsia. They can transfer the bacterium to their offspring as well through the cytoplasm of their eggs, and that's called a vertical transmission here. So yeah, so you can see how the bacterium will continue to spread throughout the ticks, and then it can spread to humans as well. Yeah, and the disease it causes is called Rocky Mountain spotted fever. I think it's because the ticks are common in the Rocky Mountains, but I'm not exactly sure. So symptoms include fever, headache, rash, flu-like symptoms. You can see the rash on the left side here, and the mortality rate is actually pretty high. So the treatment is of utmost importance here. So, and then doxycycline just tends to be the most effective treatment. I didn't really go into the specifics here, but I don't think we have to know too much about that, just in case, because I remember sometimes they'll ask about specific diseases caused by microbes and then how you might treat them. So I just wanted to include this treatment slide on every one of these microbes here. All right, next let's go to Mycobacterium tuberculosis. So this is a gram-positive and acid-fast bacillus, and it causes tuberculosis, as the name suggests, or TB, right, in humans. And remember, acid-fast just means that these guys have what's called mycolic acid within their membranes. So when they have mycolic acid, it kind of makes their membranes very resistant to and very strong. So remember that acid-fast bacteria often they can't be decolorized during a gram stain well because their cells are so resistant that they'll kind of keep the crystal violet stain kind of trapped in their membranes here. So M. tuberculosis, right? It's usually spread through droplets, so inhalation. So if someone coughs who's infected, for example. So once you inhale the bacteria in these droplets, they make their way into your lungs, and then they're taken up by they're taken up by phagosomes, and we'll talk about these in a second, within the alveoli. So you might have seen these pictures, right? At the end of your lungs, at the end of the branch, there's these like sac-like things called alveoli that help with the gas exchange O2 and CO2 here. But essentially, once they get there, they're taken up by immune cells, and these immune cells have what are called phagosomes, and these are just kind of, so you can see at the bottom here, the bacterium in red gets taken up into a phagosome. So it's this like vesicle within the cell through phagocytosis. And then eventually what will happen is that phagosome, what it's supposed to do, it's supposed to go and fuse with a lysosome. So if you recall, lysosomes are kind of membraned organelles that are filled with hydrolytic enzymes that break stuff down, as well as what are called reactive oxygen species. These are just chemical compounds that are very reactive and will go and destroy everything. So you can think of them as kind of like the trash compactors of the cell. So the lysosome and the phagosome will fuse, and then the lysosome's internal components get released upon the bacterium, which will break it down and kill it, and then the trash is kind of just released outside. So this is what's supposed to happen normally when the bacteria enter the phagosome. But what happens here is that M. tuberculosis kind of resists phagocytosis, or it resists, it prevents the phagosome from fusing with the lysosome through a series of very complicated mechanisms here. But essentially what this means is that the bacterium kind of just sits inside this phagosome without getting destroyed, and it can multiply and grow inside here, and kind of the infection can keep pursuing. And of course, as the infection worsens, the inflammation increases, then the bacterium can actually spread from the lungs to other parts of the body as well, even though the lungs are the primary place where the effect is noticed. Yeah, so essentially your immune system, in an effort to contain the infection and prevent it from progressing, it forms what are called granulomas. So you can see on the right here, these granuloma, these kind of heart structures, they have these immune cells containing the bacterium within the middle. And on the outside, they have other immune cells that kind of surround and then keep the bacteria contained inside here. But yeah, so these structures over time can actually calcify and harden and kind of become rigid, and they create these lesions, these like areas within the lungs called Ghosn complexes or tubercles. And this tubercle thing, this is where the name tuberculosis actually came from. So that's pretty notable there. And these this is good for containing the infection, right? But the bacteria, this allows them to persist within these tubercles for extended periods of time, which means that even if you get infected by TB when you're young, you can kind of, right, the bacterium kind of persists inside of your lungs and can reemerge. So it'll kind of stay there. It can actually stay there for your entire life as well, even after treatment. So yeah, so TB kills around 2 million people a year, so pretty significant disease. Like I said, it spreads by droplet transmission through usually sneezing here, where it gets and then it gets sucked in through the lungs. Then symptoms include cough, chest pain, fever, chills, sweats, all the standard stuff. And right, we mentioned that the Mycobacterium, this genus, so M. tuberculosis, is part of the Mycobacterium genus, this group of organisms. So it can be difficult to treat because they're acid fast, right? Excuse me. So like I said before, this means they have mycolic acid, which makes the cell surfaces very resistant to decolorization and overall less permeable and very strong and resistant. So since this is a challenge, what we can do is use isoniazid. I don't know how to say these drug names, but yeah, isoniazid, I think. And this is used to inhibit the synthesis of mycolic acid. So essentially that'll weaken the membrane of this guy. And that's, yeah. And that'll kind of mess with the bacterium and then make it a lot less resistant and less hardy. Yeah. Okay, so we're moving on to Mycobacterium leprae. So this is a second bacterium within the Mycobacterium genus, just like the tuberculosis one. So this one is what we call so it usually stains gram-positive, but it can also be called gram-ghost. And this is because it doesn't really stain well because of the acid-fast nature. So sometimes when you do the gram stain, it'll kind of just show up clear instead of staining purple or pink. So that's why I said. And it causes leprosy and the more common name now is Hansen's disease, because leprosy kind of has a negative connotation associated with it. So usually people call it Hansen's disease nowadays. And it can spread through humans from this other reservoirs, which are armadillos and some species of squirrel as well. So this is why, I don't know if you guys have heard, but like in Oklahoma, some people say like never touch a dead armadillo. This is why, because like armadillos can actually give you leprosy or Hansen's disease. So yeah, so again, this one is also obligately intracellular, which means it can only survive when it's within host cells. And you can actually see that physical manifestation of that because it has a very reduced genome. So in comparison with other similar species such as M. tuberculosis that we talked about, it has a loss of around 2,000 genes. So it's kind of reduced its genome so much that it can't depend only on itself. It has to depend on the genes of the cell that it's contained within. And that's due to coevolution over kind of thousands, tens of thousands, however many years, just over time. That's how it's developed here. Yep. So amyloid spray is also inhaled and taken up through by breathing. It kind of makes its way through the nose mucosa, right? The mucous membrane within your nose. And then what it can do is it can infect two types of cells. The first is Schwann cells. So I think you guys have gone over the anatomy of a neuron, right? But when you have a neuron, right, the cells that make up your nervous system, they have these what are called these myelinated sheath areas here. So essentially, these are cells that kind of surround and they help the electrical signal from the neuron kind of travel along the next cell really, really fast in what's called saltatory conduction. That's not important. So yeah, so essentially, these help the signal go really fast within your nervous system, and these are called Schwann cells here. And they can also infect dermal histiocytes. These are macrophages. Macrophages are just a type of immune cell that do the phagocytosis thing where they swallow up, right, the bacteria and break them down. And these are just the macrophages that are located within the skin. So the infection of the Schwann cells can actually lead to significant nerve damage because remember, these Schwann cells help conduct the signal within your nerves very fast. So when you start breaking down these cells by getting infected here, that makes the signal from the neuron become much slower, and it causes what's called demyelination, where this layer kind of starts to go away. These little bumps here, right? They start to go away, and this can lead to numbness and paralysis because the speed of the neuron interacting is slowed down a lot. And then, of course, the infection of the skin can cause various lesions, inflammations, deformations, even in the hands that you often see, which is characteristic of Hansen's disease, as you can see on the right side here. And then you also get some of these discolorations and these irritated patches of skin. And that skin can be thick and dry. And then, of course, if you get if that infection kind of progresses towards the face and into the eyes, then you can have actually blindness as well because the neurons that connect to your eye are kind of getting slowed down and messed up there. So so these are just names of drugs that right, I'm not going to go in, or I mean, I don't even know specifically why, like these are the ones that have been used. And like I said, a lot of treatments of using specific drugs, right, the mechanism isn't even fully known. We just know that these drugs happen to work the best through repeated use and over decades of experimentation. So we use rifampicin. This is an inhibitor of RNA polymerase, right? So that'll stop the bacterium from doing transcription and transcribing its genes. And then we also have dapsone, which inhibits what's called dihydrofolic acid synthesis. And this dihydrofolic acid, it's a precursor or something that's necessary to create a nucleotides, which means that if you use dapsone, then the bacterium is going to have a hard time making new mRNA, and it's going to have a hard time making new DNA to replicate as well. And then clofazimine is another drug that's used here. Okay, Helicobacter pylori. I really need to check the pronunciations before I make these slideshows, but please bear with me. And I guess you can search them up if you're curious. But this one is a gram-negative helicobacterium, so it has this unique helix shape, and it's known for causing gastritis or stomach irritation and stomach ulcers, which is where you get these kind of open infected sores within your stomach that are very painful. So this one is unique because it's colonized humans. It's colonized the vast majority of the human population. Almost everyone kind of has these, but they're kind of just sitting there, and they can actually be helpful in some ways. It's only, like I said, when our immune system gets messed up or you get too many of these bacteria, that's when you start getting these negative symptoms here. So once you get infected by the bacterium, and it's kind of mysterious how exactly people get infected here. So I didn't really include anything there because it was a little bit unclear in the literature. But So it what it does is it'll burrow, it'll get to your stomach, it'll burrow into the layer of mucus that is kind of on the outside of your stomach, right? That mucus layer helps protect your stomach cells from the hydrochloric acid that's within your stomach. So it burrows using its flagella, gets into that mucus layer, and then what it does is actually, right, these bacteria actually pretty much die in like pH around pH 4, but your stomach is pH 3. So it's 10 times more acidic than the death pH of these bacteria. So they need some mechanism to be able to withstand that, right? So what they do is they produce this enzyme called urease. What it does is there's a lot of urea kind of present within your stomach naturally. This urea is just like it's a compound made of carbon, oxygen, and nitrogen. So this urea, the urease will break it down, and when it breaks down, it gets turned into ammonia or NH3 and CO2, carbon dioxide. And if you know anything about ammonia, right, you know that it's pretty basic. So so since you have this basic substance being produced all around the bacterium, it creates this kind of bubble of a reasonable pH because the acidity of the stomach is neutralized by the basicity of ammonia. So that's how they survive here. And then once it gets there and it does this and it keeps growing, infecting, etc., the bacterium, again, it can release various toxins that essentially can kill host cells, and they can also degrade the mucus layer of the stomach. So once that mucus is degraded, right, the hydrochloric acid in your stomach can start attacking the stomach cells as well. And even the ammonia that's produced from the urease activity is actually toxic to the body as well. This is actually why, like urination, when you urinate, you're releasing a lot of the urea that's in your body because essentially I may be getting this incorrect, but I think this is right. So I believe the ammonia in your body that's toxic is converted to urea, and then that urea is put into your urine and then excreted. I think that's correct. But yeah, anyways. And then you also have, right, these are gram-negative bacteria. So they're lipopolysaccharide, right? That outer layer of on the outer membrane, you have that sugar layer sticking off like a little hair on the outside. They can also induce an immune response, which just makes the inflammation worse and the infection more serious here. So again, right, we said they can cause gastritis. This just refers to inflammation of the stomach lining. And this is actually one of the most interesting stories I've heard in microbiology. But so H. pylori, it was discovered when this guy named Barry Marshall, so he was trying to prove that this bacterium could cause gastritis in people. So what he did is he took a liquid culture of this bacterium, and he literally just drank it and then tracked his symptoms over the next few days. And what happened is that he actually developed gastritis in like 4 or 5 days, I think. So he kind of proved that, and he actually won the Nobel Prize for his research on this bacterium. And then he cured himself using a series of antibiotics as well after a few days. So I just think that's one of the most interesting stories that I've heard recently in science. But yeah. So, like I said, the infection continues to develop, causes more irritation, stomach ulcers, and it's even been implicated in stomach cancer recently due to all the damage. And I think there's a whole other pipeline of different factors, but I didn't really get into that here. And treatments vary, but they usually include a series of antibiotics just so that they don't get resistant to one of them. You kind of attack them from 2 different angles at the same time. And you can also use what's called a proton pump inhibitor or an H2 inhibitor that just reduces the acid production within your stomach so that your stomach cells get less irritated. Okay, finally we have Microcystis aeruginosa. This is a cyanobacterium, so very different from the ones we've been seeing so far. So these guys they're not going to infect or enter your body unless, I guess, you like drink like pond water or something. But they're a cyanobacterium, which means that they're photosynthetic and they're gram-negative. and they're often involved in algal blooms. So this is kind of when you see like, for example, like on Cassidy Lake, right? You see that like gunky green layer that kind of grows on the top of the lake, right? That's an algal bloom. It's where you get an extreme overgrowth of algae slash cyanobacteria, and they kind of colonize the surface of a body of water. And this is actually very, very bad for a number of reasons that we'll get into. But for this specific species here, Amaruginosa, it can produce toxins, which means that if there's a lot of blooms, then the water in that area is going to get polluted with several toxins that can cause disease, which is bad for humans. It's bad for the local wildlife there, as well as the other microbes within the lake water or the whatever water you're in. It's mostly found in fresh water. So blooming, one of the most significant reasons for this overgrowth of algae, as you can see on the right here, is due to what we call eutrophication of water sources. So eutrophication is where an environment where water gets really enriched with a lot more nutrients than are normally there. So for example, you can get runoff from like agricultural industrial sources. So you get a bunch of chemicals from let's say like fertilizers from a local farm or something, and that will get kind of go into the water. Then those fertilizers have a ton of nutrients within them so that they can give food to the plants, right? But now they're in the water, so now there's an overabundance of nutrients which the algae, or sorry, the cyanobacteria can use to grow a ton. So that's one of the main reasons for this. And in fact, Microcystis aeruginosa is shown to be very good at utilizing glyphosate, which is this is actually, I think, if you take Dr. Bishop's Africa seminar, then you will know what this is. But essentially, it's a common herbicide, or it kills off weeds, but it's a very good source of phosphorus for M. aeruginosa, so it can grow off of this, and it's one of the main causes for these blooms. So the specific toxin that this cyanobacterium produces is called microcystin, and it's what's called hepatotoxic, or toxic to the human liver, and it can also have negative effects in various animals as well. One of the issues with it is that it's very stable and resistant to breakdown even after boiling water. So even if you boil your like water to drink it or like purify it, right, it's still going to be there and it can still have effects. So and I believe when I was reading on this, I believe there weren't any like deaths associated with it, but it did contribute to significant liver damage, so it's probably still, pretty very, very, very not good if you ingest it. So and the way it works is that it inhibits phosphatases. So these are enzymes that are found in pretty much every organism. What they do is they remove phosphate groups. They remove like phosphorus P groups from proteins. So usually within the body, you have what are called signaling cascades. So it's essentially like at the cell, you remember you have these like receptors, right? So on the cell surface, once a receptor is bound and activated, it kind of needs to go through a chain of events to kind of induce some change in the cell. And usually that's in the form of gene expression. So you want something at the surface to impact something that's within the nucleus. So you want to like change the DNA and not change the DNA, but I mean, we'll probably talk about this in the future. Yeah. So it does that by activating a series of proteins, and then that will make its way all the way to the nucleus. And usually that happens by adding phosphate groups. So you can have protein 1 can add a phosphate onto protein 2, which activates it, and then protein 2 goes and adds a phosphate to protein 3, which activates it, and over and over and over, and then eventually the last protein will make its way into the nucleus and then cause some change there to change the overall activity of the cell. Sorry, that's kind of a tangent, but so these phosphatases, they are the deactivators. So once the proteins are activated with a P, they take that P off. So when you inhibit these things that deactivate the system, then the system's going to continue to be activated. So that means that like our cells are just going to keep doing like things that we don't want them to do, that we want them to stop doing, and that's why this is a big problem here. And again, exposure for humans can occur from drinking water, and it can even occur through ingestion of supplements that contain algae, as well as fish that graze on the cyanobacterium. And algae are technically separate from cyanobacteria, but in the past they were kind of classified as the same. So I'm just saying algae to refer to these bacteria here. So the cyanobacterium, they raise the pH of the water as well, and which is very, of course, that'll change the environment and be negatively impactful to certain life forms that are within that water. And they can also deplete oxygen. They use up all the oxygen, which can also kill off more life. And the way to mitigate or kind of stop these blooms from happening, right? First, you need to redirect the runoff from nearby sources, prevent that fertilizer from getting into the water. And then cyanobacteria, right? They're generally dependent on phosphorus sources. So if you can find a way to deplete phosphorus by using a series of chemicals, that can also be effective. You can also introduce natural predators of cyanobacteria, such as mussels and plankton. These guys will just eat up and reduce that cyanobacteria population. And of course, you can manually remove some of the cyanobacteria from the surface with nets. And you can also use chemicals such as copper sulfate and hydrogen peroxide, which are toxic to these bacteria, and you can kind of kill them this way. And that's it.